Welcome back to my next class. So we have been talking about active sites, enzyme, catalysis, this, that, and that. So now quickly look at when we say activation in enzymology, what do we really mean? No, I said we'll talk about activation and repression phenomenon. So what do we mean when we say activation? Now, activation of an enzyme refers to the process of increasing its activity, often by binding a regulatory molecule called an activator. I hope we are taking notes. Activation of an enzyme refers to the process of increasing its activity, often by binding a regulatory molecule called an activator. While repression of an enzyme now means decreasing its production at the genetic level, essentially turning off the gene responsible for its synthesis, usually triggered by the process of a high concentration of the end product of a metabolic pathway. Both mechanisms are key ways cells control their biochemical processes. So when the body requires the catalytic activity to be increased, something called an activator is being attached, and that will help to speed up the catalytic reaction that the body requires in carrying out biochemical catalysis at that moment. Then when body does not require speed, there's a way it happens. That's why it's called a phenomenon. So it's a kind of how do I put it? When one is high, the other one has to be low. When the other one is low, something else will come up to increase it. So that of repression, the repression aspect of it. So the repression aspect of it happens in this one now. It happens at the genetic level. It happens at the genetic level where there's a decrease in production of that particular enzyme at the genetic level, essentially turning off the gene that is responsible for the synthesis of that particular enzyme, because at that point it is not needed. Usually it's triggered by the presence of a high concentration of the end product. The product that, when substrate and substrate binds to an active site of an enzyme, what happens? I told us before that products will be formed. And at the end of the product and an enzyme will be formed, and the reaction continues until it is completed. So when there's enough product in the system, the repression phenomenon comes into play, where it is being handled at the genetic level, where there will be no synthesis of that enzyme at all. Yes, we said this is triggered by the presence of a high concentration of the end product of a metabolic pathway. These two mechanisms, they are key ways that cells control their biochemical processes. Welcome back. So still on our activation and the repression phenomenon. So under activation, the mechanism is an activator molecule binds to a specific site on the enzyme, causing a conformational change that exposes the active site. Okay, making it more accessible to the substrates. That is how it makes it fast. So instead of substrate to take time to want to know where to bind, and so this activator helps it to make that enzyme active site to be more it causes it to change, and it makes it to be more accessible for the substrates, thereby leading to acceleration in enzyme catalysis. Okay, example says some enzymes require the binding of a metal ion like calcium for activation. Yes, calcium ions also helps. It's an activator now. Yes, not an enzyme. Yes, but if you look at most enzymatic activities or whatever, you see calcium ion being present. Then under repression, we have the mechanism. When the end product of a metabolic pathway accumulates, so when it accumulates, it binds to a repressor protein, which then prevents the transcription of the gene encoding the enzyme needed for the first step in the pathway, effectively reducing the enzyme's production. I think I've said it before. Okay, example. Now, the lactoperon in bacteria, where the presence of lactose induces the production of enzymes needed to break down lactose, while the absence of lactose represses the production of these enzymes. Okay, so activation directly affects the activity of an existing enzyme molecule, while repression controls the synthesis of new enzyme molecules by regulating gene expression. What is the feedback mechanism now? Repression is often considered a feedback mechanism where the end product of a pathway inhibits its own production. The product itself, you know, enzyme activity starts from where the substrate binds to the enzyme active site to give birth to the product, and the product now to give birth to product and the enzyme, and another cycle continues. Now the end product now, the end product itself, where the end product of the pathway now inhibits its own production now by repressing the enzyme responsible for the first step. That is feedback mechanism, which is the repression. Then we also have what we call enzyme induction. Enzyme induction is the process of increasing the activity of enzymes. Enzyme induction is the process of increasing the activity of enzymes in response to a stimulus. It's different from activation, so that we will take notes. Activation, where we say activator binds to the enzyme's active site to make the active site more accessible for substrate. Now we're saying enzyme induction is the process of increasing the activity of enzymes now in response to a stimulus such as a chemical or drug. This increase in activity can change the effects of the stimulus on the body. How does it work? A molecule such as a drug now, for example, binds to a repressor and prevents it from blocking the transcription of a gene. Repression is supposed to block the transcription of a gene so that enzyme will not be produced at all. Now, a molecule such as a drug now binds to the repressor itself and prevents it from blocking the transcription of a gene. This allows for the increased production of the enzyme. The enzyme can then promote the synthesis of more enzyme protein. Examples of drugs that induce antibiotics such as rifampicin, okay, those are examples, even antiretrovirals too, okay. They'll have inhibition. Enzyme inhibition. So enzyme inhibition is the reduction. It is the decrease in what? In reaction rates. Decrease in reaction rates. And at the end of the day, these are being carried out by what we call enzyme inhibitors. We have different types of enzyme inhibition. We have the competitive, we have the non-competitive, competitive, non-competitive, uncompetitive, mixed, irreversible. Those are examples of enzyme inhibition. A competitive inhibitor and substrate cannot bind to the enzyme at the same time. A competitive inhibitor and the substrate cannot bind to the enzyme at the same time. Note, often competitive inhibitors strongly resemble the real substrate of the enzyme. Yes. While for that of a non-competitive inhibitor binds to a site other than where the substrate binds, another place. Now the substrate still binds with its usual affinity, and hence the chemical reaction remains the same. For uncompetitive, an uncompetitive inhibitor cannot bind to the free enzyme, only to the enzyme substrate complex. So when there's an enzyme, when the substrate has binded to the active site, that is when a non-competitive inhibitor can bind. Hence, these types of inhibitors are most effective at high substrate concentration. In the presence of the inhibitor, the enzyme substrate complex is inactive. This type of inhibition is rare. Then we have the mixed inhibitor. A mixed inhibitor binds to an allosteric site, and the binding of the substrate and the inhibitor affect each other. We have the irreversible one, where an irreversible inhibitor permanently inactivates the enzyme, usually by forming a covalent bond to the protein. So those are the types of inhibition that we can have. We mentioned about, I think, 5 of it: competitive, non-competitive, uncompetitive, mixed, and irreversible. What are the functions of these inhibitors? In many organisms, inhibitors may act as part of a feedback mechanism. If an enzyme produces too much of one substance in the organism, that substance may act as an inhibitor for the enzyme at the beginning of the pathway that produces it. causing production of the substance to slow down or stop when there is sufficient amount. This is a form of negative feedback, just like we talked about repression mechanism. So that is how it is. So major metabolic pathways such as the citric acid cycle, they make use of this mechanism. Okay. So see you in my next class.